Uh, Hello everyone. Uh, my name is
Hello,
Sho.
sir.
Yeah, hi. My name is Shomo. I'm a third year PhD student in ISC. I'll be taking this uh, live session classes for this particular course for bioinformatics. I think there is another person who is taking these live sessions. We will try to uh, kind of discuss different things or like distinct things in these two sessions. But we'll see. But the idea of these sessions are that it's a 2 hour session. It's doubt clearing. So if you have any questions, you're free to ask anytime. Uh, Other than that, the idea would be to uh, that like so we are given previous year questions from this particular course, and we solve those questions in this live session. So generally 10 to 20 questions for each session, depending on the uh, topics that has been taught for that particular week. Uh, this will help you in doing assignments and like for the final exam when it will happen. So how I take these sessions are basically so since it's a 2 hour session, so the first hour or so, or 45 minutes or 1 hour, I try to discuss, kind of give a recap of what has been taught for that particular week. Uh, like I make a presentation and I kind of give a recap of whatever topics has been taught. And after that, we discuss those questions that I was talking about. So, yeah, so generally that is how I do. So if you have any questions, any kind of questions, you can either either write in the discussion forums or come to the live sessions and ask, or you can ask me uh, sorry through my mail ID as well. So if you have any questions, do drop an email. I'll try to get back. Okay, I think yeah, that is all I have to say in terms of the introduction. So I would like to know what kind of um, backgrounds you all are from. So if you could write in the chat box or like unmute yourself and tell me like what kind of.
Sir, my name is Sadika Gurdel,
Yeah.
and I'm studying in BSc Biotechnology second year
Okay.
from Gurugram University.
Okay. Okay. That's good. That's good. So how many like I'm hoping a lot of people are coming from biology backgrounds. Is there anyone that is coming from a different background other than biology? Okay, seems not. Okay, that's good then. Um, okay, so this course will mostly focus on different bioinformatics applications. So we'll look into different databases, different algorithms that are used from this field. But the application of those algorithms can be vast. So we will not be able to discuss every application possible because there can be a lot. So uh, there will be a lot of applications we do not discuss, but those are applications of the algorithms that we will be studying. Just to remind you that this is a very vast field. There's a lot of topics, lot of applications, lot of algorithms and so on. So just do not try to limit yourself to what you are studying here. Only like if you are really interested, please go back and like kind of read up upon the work that has been done on those kind of things, mostly research papers. So because the field is very vast, we'll not be able to cover each and everything possible. Right? Okay, so I think we can start now the session. Okay, and we share my screen. Okay. I hope you can see my screen. Can anybody please confirm? Yeah, sure. Thank you. Okay, so this is an introduction. So the first week is an introductory week. Not much has been taught in the whole duration of the week, but some overviews have been given in terms of what kind of things we'll be studying, what kind of applications we'll be looking at. Just to give a small heads up, since this course is a rerun course and it's kind of a little old, uh, so there will be a lot of things that has happened. So I think this course is around 6, 7 years old. So after that, there has been a lot of improvements in different bioinformatics algorithms and applications. So a lot of a few things that we'll be discussing as a part of the course syllabus. Some of these those things are probably not very well used or very well maintained because the newer softwares have come, newer techniques have come, so the older ones have been discarded or like not maintained. So in some cases, it is possible that we'll be studying something here, but if you go back and look at the actual applications, it is not probably well maintained or something like that. That is possible, just to give you a heads up, because this course syllabus is a little bit older. So today we'll be discussing very briefly about DNA structures because a lot of the data that that bioinformatics handles is coming from molecular biology level data. So it can be DNA sequence data, it can be RNA data, protein data, and so on. So a lot of the data that will be handling are coming from molecular biology data, like mostly 80 to 90% of the data. There can be other data types as well, say for example clinical data or phenotype data and so on and so forth. But mostly we'll be focusing on DNA, RNA, protein, and so on, molecular biology data. So today's session, I'll just try to give a primer about what are the type of like kind of what are basics of DNA and DNA structures and so on, and basically how we get data from that. Right. Okay. So just a very simple overview. So uh, we all know the central dogma of biology, which is DNA. It replicates itself, DNA transcribes itself to RNA, and finally RNA is converted, translated into proteins. Sorry. Yes. Yeah. I think I should log in through my phone so that I don't have to go back to admit people. Okay. Um, yeah. So DNA is the genetic material, and it is composed of basically two polynucleotide chains that are antiparallel to each other, and they're twisted to form a double helical structure, where you have these set of ATGC base pairing, where we have two types of nitrogenous bases in this DNA, which is purine and pyrimidine. So adenine binds with thymine with two hydrogen bonds, guanine binds with cytosine with three hydrogen bonds, and the two antiparallel strands of the DNA are always complementary to each other. So if you have A in one strand, there is going to be T in the same position in the other strand, and same for G and C and so on. Right. So and this bonding between complementary strand bases is done through hydrogen bonds. And basically, as I was saying, A and T will have a double hydrogen bond, and C and G will have a triple hydrogen bond. And this base pairing is very specific. If this base pairing does not occur correctly during, say, for example, replication, uh, there has been some error in replication and this base pairing is not occurring properly. What happens is the bases flip out of these double helical structures, and there are repair mechanisms inside the cell which kind of um, find these sites where this hydrogen bonding is not happening properly, and they kind of repair these sites because the a correct base has not been put there. That is why the hydrogen bonding is not correct there. So the structure of the DNA is uh, very important in terms of everything. So, so we will be seeing some applications of how to predict structures, but generally DNA structure or protein structures are experimentally not predicted, determined through X-ray crystallography and techniques like that. But bioinformatically as well, if you know the sequence of the protein, or you know the sequence of the DNA, you can predict if there is a minor groove or a major groove in that region for DNA, or how the secondary or tertiary or quaternary structure of the protein would look. So structure prediction from the sequence is something that bioinformatics is heavily involved in. So we will be seeing in further classes some examples of that. Now, coming back to DNA, so yeah, as I was saying, the central dogma of biology, DNA replicates itself, then it is transcribed into mRNA, and that mRNA is translated further into proteins, which are basically long chains of amino acids. Right. So yeah, just to give a visual representation. So this is the double helical structure here. So one strand runs from 5 prime to 3 prime, the other strand runs from 3 prime to 5 prime, anti-parallel direction. And this is how the complementary base pairing looks. So if you have a G and C, you have three hydrogen bonds. If you have a A and T, there you have two hydrogen bonds. Here, this is not shown that accurately, but I hope you can get the point. And these are very specifics of the structure of the DNA as well. So this one groove is around 3.4 nanometer, and around and one the distance between two bases is around 0.34 nanometer, because in a single turn there are around 10 bases, and it's around 3.4 nanometer or 34 angstrom. So for each base, the distance between a pair of bases is around 0.34 nanometer. Right. Now, in terms of storing and copying information, it is done through these different base pairs. Sorry, guys, I keep on stopping. Oh, sorry. Am I not audible? Am I audible now?
You're sir.
Okay.
So we can hear you.
Okay. Thanks. Yeah. Uh, yeah. So, DNA in itself stores information in terms of the sequence of the bases it stacks up upon one another, and the complementary base pair uh, matching that happens when in the 3D helical structure. So the storing of the information here is basically the sequence of the nucleotides that are present. So the sequence is itself the information that confers to it. So a particular sequence will code for a particular mRNA that will code for a particular protein. So the information is stored in the sequence. Also, something that we do not discuss here, but it is quite important. So for eukaryotic uh, organisms like human beings, we have something called DNA modifications like DNA methylation or acetylation, because our DNA is packed around histones. So we have a region of the genome which is euchromatin, where basically the packaging is a little bit loose, and there is another portion of the genome called heterochromatin, where the DNA is very tightly packed and not very accessible to transcription factors or DNA replicating proteins and so on. So depending on the region of 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 the DNA, it can have different methylation patterns on the histone proteins. So depending on the methylation patterns of the histone proteins, the transcriptional and the translational mechanism work. So if it is highly methylated, mostly the region of the DNA is silent in terms of transcription. Transcription does not occur there much. If it is less methylated, it is more active in transcription and so on. So information is can be both. So this is the genetic information, the sequence of the nucleotides that is present. There can be epigenetic information that is stored as well, which is the methylation patterns and other uh, DNA binding proteins and so on. So yeah, mostly two types of information that can be stored. And in terms of copying information, as I was talking about, the central dogma of biology, which is basically replication. So I don't know if you have studied or not in your maybe in your school syllabus also. So there is the semi-conservative nature of DNA replication. So basically, if you have two strands of the DNA, at one point of time, the strands break apart, and one strand acts as a template for the new daughter strand. So in further generations, you do not have like a whole new copy of both the strands. You have one copy of the parent strand and one newer copy of the daughter strand. So in terms of copying and storing, DNA works basically in this manner. There are a lot of details involved which we are skipping, but just to give you a primer about how information is stored and copied. Okay, uh, yeah, I'll not go into this again. Basically, it's the base stacking I was talking about: A and T versus G and C through hydrogen bonds and so on. So, yeah. Yeah, so just to tell you the visual difference in terms of the nucleotides. So basically, these are the set of nucleotides are the building blocks of the DNA. So there are three components of a nucleotide. One is the nitrogenous basis, which is the ACG of T, adenine, guanine, cytosine, methionine, and then you have the pentose sugar backbone, and you have phosphate bonds that connect two different nucleotides. So in the backbone of the DNA is made up of a phosphate sugar backbone, and then on top of that you have nitrogenous bases which stack up on one top of one another. Now, the next level of information comes at the genetic code level. So we were talking about replication and DNA. So the next step of information comes from mRNA, which are DNA when transcribed forms an mRNA. You have enzymes like RNA polymers and those kind of other machineries which convert a DNA, stretch of a DNA to a functional mRNA, which can be translated into proteins. Right. Now, in our case, as I was saying, in eukaryotes, the coding region is not continuous. You have intronic regions in between exonic regions, which basically do not code for any protein. Previously, it was thought to be junk DNA and it is not required. Intronic regions are not very important, but recently there has been studies which show that variants in the intronic region of the DNA can affect the exonic regions as well. So it's not junk DNA, but a lot of the DNA that we have is not used for protein coding. So the structure is basically you have a set of exons, which are your protein coding regions of the DNA, which are interrupted by introns. So what the mRNA during transcription happens is basically uh, it it transcribes only the exonic regions and skips the intronic region. Itself is a very fascinating mechanism. We will not go into that. But yeah, so we do not in prokaryotes, it is possible to transcribe an entire stretch of the DNA, but in humans and other eukaryotes, we have regions of the DNA which do not code for any protein, so those cannot be transcribed. But after transcription, you have a long stretch of mRNA, which actually fully codes for the protein. Now you have, for humans, you have removed the introns. For prokaryotes, you do not need to do anything. But like after that, after removing the mechanism is called splicing, where you cut apart the introns and move it away, and you join all the exons together. And after that, you have other mechanisms where different proteins come and bind to the mRNA and give it more stability. Those are RNA binding proteins and other kind of proteins that are there. So there's a detailed mechanism in itself, but the idea is that you have now a long stretch of mRNA which codes for protein. So how is the protein coded? So the protein is basically a long sequence of amino acids, as in for DNA, the sequence of the amino acid is important, which makes up the protein. So there are total 20 amino acids possible, and different combinations of these amino acids make up the whole protein. Right? Now, in the from the mRNA to the from mRNA to protein, this process is called translation. So in translation, what happens is basically we take uh, we try to decipher the information from the mRNA in in uh, in groups of three triplets of base pairings. So the idea is that three nucleotides together code for a particular uh, amino acid. So that is what is known as the genetic code. So you have a sequence of these A, T, G, C. In case of RNA, you won't have T. Instead of thymine, you'll have uracil. But in terms of the base pairings, the sequence of these bases is divided into blocks of three, and uh, these three bases together determine a particular amino acid. And to start the 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 function the coding of a protein, we need to start with a start codon, which is always AUG, which is a methionine, which codes for methionine. So this is the genetic code. So our genetic code is basically different combinations of these sequences in the mRNA and what are the different amino acids it can code for. Notice there are some degeneracy in the genetic code, so there can be multiple different combinations of this code coding for a similar amino acid. And also there are some examples like tryptophan, which only has one codon. Like there's only one codon that codes for tryptophan, and there is a lot of, say, for example, for proline, there are four codons which are which are coding for proline, but in case of tryptophan, it's only one. Right. And it is represented in this manner. You take the first letter from here, you take the second letter from here, and you take the third letter from here, and then you make all the combinations and you see which are the possible amino acids possible from these all combinations. So it starts with the methionine, which is the AUG. Then it checks the codons three at a time. Uh, Codon is basically three uh, nucleotides at a time. So it takes three nucleotides as one codon at a time and checks, and it so that there is the ribosome inside the cell which comes and binds to this mRNA, and it actually the ribosome and the transfer RNA and other RNAs involved, they read this particular genetic code and they bring the amino acid from the environment and then put it in a chain of amino acids, ultimately forming the protein. Right. So, uh, yeah, quite interesting in that manner. Okay, so after that, another important concept that comes in terms of the codons itself is the open reading frame. So the open reading frame is basically a portion of the DNA sequence which does not include a stop codon. So basically, the whole coding sequence of a particular mRNA is, or a DNA sequence is called the open reading frame. And so basically, you have this set of three nucleotides which are your codons. Uh, uh, which codes for an amino acid or it codes for the termination of protein synthesis. And uh, total, as I was saying, there are 64 different codons, 61 specify an amino acid. So out of 64, 61 as coding for 20 different codons. So you can understand the kind of degeneracy that we have here. And the three different codons are used for stop codons. So generally, that is what is called as the open reading frame. It always starts with this particular AUG, but the stop codon is not considered a part of the open reading frame. The start codon is, right. So a slight trick here to remember that you always have the start codon inside the open reading frame, but the stop codon is actually outside it. So, yeah. As I was saying, that it basically is the region of the DNA that the ribosome is reading to make the protein. So that is the open reading frame. Okay. Next, we have a couple of important uh, examples in terms of not examples, rules you can say in terms of the DNA pairing mechanism that we know. These are very simple in terms of understanding the rules. So first is the Chagas rule. It states that sorry. Yeah. So in sorry. In any organism, the amount of guanine is equal to the amount of cytosine, and the amount of adenine is equal to the amount of thymine, which is intuitive because your adenine will always pair with thymine and your cytosine will always pair with guanine. So the amount of adenine should be equal to amount of thymine, and similarly, amount of guanine and cytosine should be equal. So, and also this also extends to the fact that the total number of purines, which is adenine and guanine together, should be equal to the total number of pyrimidines, which is thymine and cytosine together. So, small numericals can come in terms of Chargaff's rules in your exams or your assignments. So, yeah, I think you can solve these things. Uh, yeah, and basically the next two are basically extensions of the first thing. First thing itself. So, in a double-stranded DNA molecule, the A and T percentages are equal, and G and C percentages are equal. And the other rule is basically that A and T is equal is valid for both the DNA strands. So if you take a strand of DNA, ideally in that whole stretch, not the complementary together, just one strand of the DNA. Ideally in that stretch, the A and the T and the G and the C should be roughly equal. That is also part of kind of Shalinoff's rule, but that is not kind of hard-coded rule. The hard-coded rule is that A should be equal to T and G should be equal to C. Right? I think I missed this here, one thing. So you have this set of nucleotides. You have these purines, pyrimidines. So this is the whole structure of the DNA in terms of the bases and the sugars and the phosphate bonds. So just to keep one thing in mind, that purines are having two circular rings, adenine and guanine, and the pyrimidines are having a single circular ring, which is cytosine, thymine, and the version of uh, unmethylated thymine is basically uracil. So all these three are pyrimidines and these two are purines. The one thing to keep in mind is uh, uh, the ring structure. Basically, you have double rings in case of purines and single rings in case of pyrimidines. Okay, that might have missed this. Okay, okay. Now I think this is something that has also been discussed by Sir in the sessions as well. So how do you calculate if you're given a set of all this information, so you are given that this is the amount of adenine, this is the amount of thymine, guanine, cytosine, and so on, and you are told that you are given this is the sequence you want to find what is the stacking energy. So this base stacking that is happening in the double helical structure, this is happening because this is thermodynamically feasible and thermodynamically like it's good for the cell. The amount of energy that is going into the stacking is somehow useful for the cell. So if it is thermodynamically feasible, there should be a delta G associated with it, which is a standard free energy associated with it. So if you're given a string of DNA nucleotides and you are asked that what is the what sorry, is anyone saying anything?
Sir, I was asking
Yeah.
that why it is good for the base stacking energy to be a certain amount, why it is good for the cell.
No, in terms of it's good for the cell in terms of uh, the the energy that is getting released. So we are talking about the delta G, right? So if your delta G is negative, it means it is thermodynamically feasible. And for the cell to have a structure of DNA where if you stack one base on top of one another. That is a thermodynamically feasible exercise, and it also brings stability to the structure of the DNA itself, right? So remember how the DNA is formed, right? So you have a phosphate and a sugar backbone. The phosphate is negatively charged, and sugar is mostly not charged. And you have another set of these nitrogenous bases, which is also kind of negatively charged. So you have a lot of charges. DNA is a negatively charged molecule as a whole. It is not a neutral molecule. So if you do not have it in a stacked manner, if you do not have it in a helical or in a ladder kind of a manner, if you put them all together in without any stacking or without any kind of uh, any kind of manipulation to the structure itself, you just put all the DNA together, it won't remain like that, because there is a lot of electrostatic interactions, a lot of other forces that are involved, because DNA is such a complex molecule that it won't the structure of the DNA will not be stable. So the best stable structure is the double helical structure, evolutionary wise, because after so many millions of years of evolution, nature has come to that, like kind of to the idea that stacking of the bases should be is feasible and is basically very stable for the structure of the DNA and the charges of the DNA as well. I hope that answers your question. But yeah, if you have anything.
Yeah, so basically to make it stable for the
Yes,
next
yes,
addition
yes,
nucleotide.
yes, yes, yes. So if you have a very highly charged molecule with such a long like sequence, it is ideal to have it packed in such a manner that you dissolve these charges and kind of have a stable molecule, so that, because again, this is a genetic material, right? It has to be the most stable molecule inside your cell so that it can propagate to the next generation. So stability of the DNA is of utmost importance, and through millions of years of evolution, we have come to this stage where yeah, this stacking is kind of the perfect conformation that we can have. Okay, so I think we have a few questions. Okay, so A and T double bond is basically due to the structure of A and T itself. Let me see if I have it here.
Yeah.
Just let me, I think I have a better pictorial representation. Mm, yeah, I think this would. Yeah, let me see. Oh, I think both are guanine cytosine. Just give me a minute. Yeah, you see this NH bonds, these amine bonds that we have. These amine bonds actually are forming these hydrogen bonds. So you have this NH2 here, NH here, NH here. These forms a bond with a negatively charged molecule, mostly oxygen. In these two cases, the double bonded oxygen, or in this case, the single bonded nitrogen. This is for guanine and cytosine. Similar is a structure for adenine and thymine. So you have the kind of the opportunity to do three bonds or two bonds, depending on the structure and the chemical composition of the molecules itself. So I hope that answers the question. Okay. Somebody asked about asked about Shargaf's rule one more time. Shargaf's rule is quite simple. So let me go back here. Yeah, the idea is that in the whole of your DNA, you have a set of adenines, you have a set of thymine, guanine, and cytosine, all kind of distributed all across the DNA. The idea is that your adenine always binds with thymine and your guanine always binds with cytosine. So the amount of guanine should be equal to the amount of cytosine. The amount of adenine should be equal to the amount of thymine. And since A plus T plus G plus C is constant, you can derive this that the total of adenine and guanine, because A binds with T, so and G binds with C, the total of adenine and guanine should be equal to the total of thymine and cytosine. So it's quite simple. Just keep the idea in mind that what binds with what. So A binds with T and G binds with C. That is, I think, should be enough. Sorry. Yeah. Okay. Yeah. So yeah. So if you are given this as a set of as a sequence with a set of nucleotide n with a length of the sequence being n, and you are asked to calculate the base stacking energy. The idea is simple. So one base stacks after one another. So the base stacking energy is calculated in pairs. So if A is if C is stacked on top of A, the stacking energy will be something. If C is stacked on top of T, the stacking energy will be something else, and so on. Depending on the energy, the delta G, as I was talking, the delta G, depending on that, you will have a set of energies that is already predetermined in terms of if base A stacks up with base B, what is the energy possible. Now, since we are calculating it in pairs, so the idea would be to decompose this into n minus one nucleotide steps. So basically, AC will be one set, CT will be one, TA will be one, AT will be one, and so on. So there will be total n minus one sequences, right? If you have the total length of the sequence to be n. Now you have already previously calculated values of delta G associated with each specific dinucleotide steps. There are a lot of biophysical studies that have been done to calculate this set of energies. We'll not go into details of that, but these are kind of well established now, right? So now after that, what you do is basically sum up all the energies of these dinucleotides possible. And since this is dependent, the stacking energy will be a function of the length of the sequencer as well. So to normalize that, we divide it by the total number of pairs that are possible, which is n minus one. So this is the total Gibbs free energy that you are summing up for all the set of pairs of nucleotides that you are looking at, and then you are dividing it by n minus 1, where n is the total length of the sequence. So simple problems might come in your assignment or your exams, so where you will be given the base stacking energies of different combinations of these nucleotides and given a sequence like this and asked to find what will be the uh, like the normalized uh, Gibbs free or the thermodynamic energy that is possible for that sequence. Sorry. So yeah, small numericals might come from this. Right. Now mm, we have talked about different types of so the molecular biology primer, basically DNA, RNA, codons, and proteins, and like base stacking energies associated with those, and so on. Now, all this information. When you're doing experiments, when you're doing wet lab experiments in the lab, uh, at large scale or at small scale, you generate these data, right? So you generate different levels of data. You can generate sequence data, you can generate expression data, you can generate different types of data, structured data, and so on, so forth. So we need databases to store this type of data. Different types of data you are generating, but you need to store this somewhere. So we have different levels of biological databases that are possible. So here I've just listed a few types. So you can have bibliographic databases where you have all the literature stored in them. So PubMed or NCBI, and PubMed mostly is the database for that, where you have kind of all of literature, publications, and literature that it stores, right? You can have taxonomic databases where it classifies maybe organisms into different classifications in terms of taxonomy. Uh, nucleic acid databases where you have DNA or RNA information present. It can be structured, it can be sequence, it can be something else as well. Then you have specific genomic databases where generally right now for in terms of genomics, what we understand is basically sequencing. In terms of whole genome sequencing, you sequence a set of samples and then you store the sequence information in a database. Or you can look at the RNA levels, the gene expression levels inside cells. So that is also another level of information that can be stored, which is the gene expression information. You have protein databases in terms of structure, in terms of function, taxonomy, and so on. And yeah, protein families, domains, so on. And you can have pathway databases as well. So these are the set of proteins that are involved in this pathway. These are the set of proteins that are involved in this pathway. And this is the overlap of the set of genes or proteins that are involved in these set of pathways. So it's kind of a graph database, and so each node will be representative of of a protein or a gene which is affecting another protein or a gene, and it becomes a whole graph of interactions. So that is also a form of a database, right? So you can have, what I'm trying to say is you can have data at different granularity across different databases. Uh, the important thing from here is to identify the kind of data that you need for your study or your work or your research, and find the appropriate databases and database for that data. Right?
Yeah.
Uh, in terms of the structure, in terms of the information that is stored in the database, it can be broadly divided. This is not a strict classification, but generally it kind of helps to understand. So you can have primary database, which has primary information in terms of sequence, in terms of structure, and so on. You have secondary databases, which actually leverage information from this primary database, and they kind of analyze some of the data and they make another database based on the primary database. So that is another type, which is the secondary database. You can have composite databases, like with some primary information, with some secondary analyzed information as well. And then you can even have another set of databases, which is basically, say, for example, organism specific database or literature information based, like PubMed and so on. So, yeah, generally, primary and secondary are the ones that we're interested in. Primary is basically very basic information. Secondary is building on that information, analyzing that data, and making another database with a little bit more uh, you have more annotations, you have more metadata on the database, more information about the primary, taking data from the primary database itself. Right? Here are the few set of databases that I am going to discuss. One second, just give me a moment. Should I go here? Yeah, sorry for the interruption. Okay, so a few databases I'm going to discuss. Uh, uh, so these are primary databases, GenBank and DDBJ. So all are open access. So these are kind of the oldest databases in terms of bioinformatics. Uh, so these GenBank primarily consists of a collection of nucleotide sequences, their protein translations, including mRNA and other ribosomal RNA and so on and so forth. So it is a kind of a mix of both primary and secondary database, but mostly primary. Secondary is very low. Uh, and it is maintained by the NCBI, which is the National Center for Biotechnology Information. So a lot of databases are maintained by NCBI. Even PubMed is maintained by NCBI. So GenBank is kind of the first database in terms of that. And the other important one is DDBJ, which is the DNA data bank of Japan. It basically receives nucleotide sequences from a host of researchers, and they assign succession numbers to these data submitters, and they kind of maintain this data. Um, Mostly this is focused in Japan, but there has been a lot of other researchers also who have contributed to DDBJ, and it is kind of one of the oldest databases for biological data, both GenBank and DDBJ. Coming to more modern secondary databases, SwissProt or UniProt QD is one of that. Um, I sincerely hope you guys take a look at the databases in your free time. Just go through it, like what it is, what things you can do. You go to there are full-on tutorials that are present for these set of databases. You can just go here. They'll give you all the information. They'll give you uh, resources and YouTube videos on how to use this databases and so on. So I sincerely hope and you go in your free time and take a look at this set. Oh, I see a lot of questions. Uh, okay, let's start from here. Why are we taking n minus one? Okay, just going back to here. What we are doing here is basically what we are trying to find the pairs of nucleotides here. So if you're given a sequence of n uh, individual nucleotides in this case, and you are asked what are the possible consecutive pairs of nucleotides from that sequence, that is going to be n minus 1 because it's a sliding window, right? So you have AC, this is one, then CT, TA, AT, and so on. So if you see this, if, if you kind of expand this, it will come to n minus 1. So you are trying to find consecutive pairs of nucleotides possible in the whole sequence. So it will be n minus 1 because you're using a sliding window of 2. Right. I think that answers this. I just put it here. Okay. Um, Base stacking energy, we have a assignment problem today only we'll solve. Don't worry about that. Uh, okay. Uh, Standard books, it's difficult for me to recommend. Probably I can take a look and come back to you next week. I personally do not follow any like standard book per se. Mostly, I uh, kind of look at papers and articles and review papers and so on. But I think Sir has mentioned one book, I think, which is written by him as well. So probably you can search for that. Other than that, I'll take a look and come back to you next week probably. I do not have any book on the top of my mind right now.
Sir, how do we practice or how do we prepare for the examination?
I think whatever is being taught in the class and the assignment questions should be enough for your examination. Examination wise, I don't think you need to worry that much. Whatever is being taught, I think that would be enough. If you want to learn more, if you want to kind of explore different avenues of bioinformatics, then probably you can take a look at different papers, modern techniques. So to be very fair, bioinformatics has come a long way. Um, right now, the hot topic for bioinformatics is next generation sequencing. That is kind of the hot topic for bioinformatics. But there are a lot of things there, like a lot of things in bioinformatics, not only NGS. I do NGS, so I am more focused on that, and like, and kind of bioinformatics has evolved to a way where this is the most hot topic right now. So if you want to learn more about that, I can definitely provide you papers and other resources. But other than that, I think there is a bioinformatics book, if I'm not wrong, less for something. Uh, I think this book, Arthur M. Lesk. Probably you can take a look at this book as well. This is kind of traditional bioinformatics in terms of a lot of details is there. It
So
is
how do we access these books?
that I am not sure. I'll see if this is this really available. Yeah, I think I just downloaded it. Uh, it's not very well parsed, I can see. Yeah, but I think all the information is there. Yeah. So, and this is a kind of an older book. The language that it is written in it. So the codes that it is written in is in Perl. So we do not use Perl anymore. Like it's kind of an obsolete language now. We generally use R, Python, and other shell scripting languages. So these books are a little bit dated, but the main idea and the concepts you will get from this book. I'm not sure if this is available freely or not. I just thought of this book because this is something that I had followed in my undergraduate and graduate time. So yeah, probably you can take a look. I'll take a look if there are other good books available or not. I'll come back to you after that. Okay. Mm, okay. Composite information contain. Okay. Composite information would be, say, for example, so the basic information would be in terms of I'm talking in terms of DNA and RNA sequence and structure and so on. So the basic information would be primary information, would be to to say this is the sequence. So for example, you are sequencing a liver cell in a human being. So you have sequenced that liver cell in terms of DNA. You know the sequence. You have sequenced some mRNA from that cell as a set of cells as well. So you have a sequence information. So these are the set of sequences that you have found in a set of liver cells for DNA and RNA. Right? That would be the primary information. The secondary information would be to analyze the DNA sequence to find variations in the DNA. SNPs, what we call single nucleotide polymorphisms or DNA variations or mutations in that DNA. So that would be, and if you find that and then you store that set of information that these are the set of variants that are present in the DNA sequence, and you keep it in a database. So that would be a secondary source of information because the primary source is present in the sequence itself. You've analyzed the sequence and said that these are the set of variants that are present inside that sequence. So that would be secondary information. Composite information is kind of a mix of both. So you have databases which have the primary information as well, but you have their flexibility to to have analyzed information data like this. Also, they'll provide you, in some cases, not in all, because this is computationally uh, very costly. So in some cases, they also provide you platforms to analyze their data in those platforms itself, in the database itself, using your codes, your maybe you can upload a couple of files and so on. So that would be an example of a composite database. So there are a lot of databases right now, all the big biobank cohorts and so on. So they kind of use this composite information. They'll have primary information. Some of it will be analyzed and given to you as secondary information, and some of it you can analyze with your own freedom, which in their platform as well. So that would be kind of composite. I hope that makes it clear.
So, sir, that means that metagenomics is also a part of the composite databases.
Metagenomics is a field in itself, but all the genomic, all the omics in general, right?
Hmm.
Probably part of, yeah, composite databases. I'll give you another example is GEO, gene expression omnibus. It's a very widely
Hmm.
used database where you have gene expression data, RNA sequencing or array data for gene expression. So in that you have a module called GEO2R where they'll give you the sequence information of the mRNAs of those set of samples. They're going to give you the gene expression, which is basically when you analyze the sequence information, you can get the gene expression information. They'll give you that as well, and they'll give you a full R console where you can write your code and um, kind of analyze that data in Geo itself. So that is a good example of a composite database. And you have a lot of these databases across omics. Across omics, like modern data, whatever you have, are mostly in composite databases.
Okay. Thank you, sir.
Yeah.
Sir,
So
how is genomics and
just a second. This will work now. I have a lot of data here. It is working, yes? Ah, okay, okay. Thank you. Okay. Yeah, anybody was asking me something?
sir, how is genomics and proteomics different from bioinformatics?
Okay. So, genomics, proteomics, transcriptomics, metabolomics, metagenomics, all the omics technologies are part of bioinformatics. So, bioinformatics is the bigger umbrella. Inside that, you have these set of omics technologies as well. There are other things as well. Omics is kind of part of bioinformatics. It's not a separate field. It's kind of a further classified version of bioinformatics. So in omics, what you're doing, so proteomics or genomics or transcriptomics, what you're doing is uh, the word omics means all, right? So basically, for genomics, you are sequencing all the DNA that is present in the cell or the sample, and you're checking the sequence of the DNA for mutations and so on. In transcriptomics, you are doing RNA sequencing or single cell RNA sequencing, array, and whatever. And what you're trying to find is basically the expression levels of the genes across different conditions. So you have a set of, say, for example, control samples, healthy samples, and you have a set of, say, for example, cancer samples. And you want to see the expression of a particular gene that you assume hypothesize might be very important for that particular cancer. Right? So what you're going to do is take this gene expression data from these two set of samples, do statistical tests on that, and say statistically if it is different or similar. So these omics technologies are basically studying a particular part of the cell. So genomics is studying the DNA sequence, transcriptomics, the transcriptome, which is the mRNAs, the proteomics, the protein sequences, metabolomics, the metabolites in the cell, and so on and so forth. So these are like studying different parts of the cell by sequencing, right? By sequencing, that is the important thing. But you're sequencing, you're finding the sequence of these long compounds or long uh, chemical molecules, and from that sequence, you're trying to predict something related to the cell itself. So that whole study, the whole field of study comes under omics, and omics comes kind of under bioinformatics because you are using bioinformatics tools, algorithms, logic, and different statistical techniques and so on to kind of tell about these these components of the cell, DNA, RNA, or protein, and so on. So it's not different. It's kind of a part of bioinformatics. Yeah. So this is like it's a very, as I was saying, that bioinformatics is a very vast field. We will not be able to cover everything in bioinformatics uh, in this 12-week course. So, yeah, if you have interest, mm, do take a look at research papers and the stuff that has been going on.
Yes.
Okay, and think there's one more question here. Basics of NGS and that skill apply machine learning as well. Oh, I am not a machine learning person. Uh, I'm not the correct guy to answer your this question. But the NGS for NGS, there is one NPTEL course as well. I think uh, the name of the course is Next Generation Sequencing Technology and Applications by Professor Ridhiman Dhar at IIT Kharagpur. And this year, that course is not running, and you can find it on YouTube or older NPTEL sites. Maybe take a look at that. That is a good course for NGS. Okay, books I'll come back next week. I'll see. These slides will be shared. Yeah, shared as in like it will be put in a Google Drive folder, and the folder will be shared with you. So for all the 12 weeks, you'll have all the slides that I'll be using. Okay, I think that is all. I'll go back. I think yeah, I'll go back to the slide. Okay, these we discussed. Yeah, I was talking about Unitrode. So yeah, these are secondary databases, but this is a very widely used database, right? This is quite old, but still very widely used. This is it is an annotated, non-redundant protein sequence database, uh, be- which means is like, say for example, both of us can, so like five of us can submit one sequence. There will be peer review, and there will be other mechanisms by which they select only one sequence out of that and put it there. So there will be no redundancies in sequences or information present. Okay. Yeah. So in initial days, this was kind of hand curated, but in later stages, I think in 1996, they supplemented this with Swissprot, and they created this TRMBL, which is this translation of EMBL nucleotide sequence databases. This contains computer annotated entries as well as human curated sequences as well. So yeah, this was Uniprot, and we have another important and widely used database, which is the PDB database, which is Protein Data Bank. Uh, this contains 3D structures of proteins. So 3D structures of proteins are very important. I hope you know that, because the structure of the protein determines which other proteins it is going to bind to, how it is going to interact with enzymes and receptors, and so on and so forth. So the structure of the protein is very important. So the prediction of the protein structure is a very important thing in bioinformatics. So prediction of protein structure, you can do it computationally in bioinformatics, but experimentally protein structures are determined using X-ray crystallography and other techniques. Uh, Recently, I think year before that, Google DeepMind came up with AlphaFold and AlphaFold 2. I think they got the Nobel Prize for it as well, where they kind of predicted a host of protein structures which have not been solvable for the past 50 years using machine learning, deep learning algorithms. So, and they got a Nobel Prize for that. So, the structure of the protein is very important, is what I'm trying to say. And PDB has the most comprehensive database for protein structures. So, maybe you can take a look at this. Okay, I think, yeah, that is all in terms of the discussion that I wanted to have. Now we'll move on to those questions. If you have any questions other than that, you can ask. Neto will move on to the previous year questions. Okay, I think we can move to the questions. I'll put the questions on the screen for a couple of minutes, or maybe 1.5 minutes. You can write the answers in the chat box, and we'll discuss. Okay. There, some older data has been stored there, so I think we need to remove that. Thanks. Okay, a lot of answers. I can see a lot of B's. RNA. There's one A here. Okay. Mm, Okay, the answer is DNA. It's kind of the wording is tricky. Methylated uracil is basically thymine, right? Uracil is present in RNA. Methylated uracil is thymine, which is present in DNA. So it's a little bit of a tricky question. So only uracil will be present in RNA, but when you methylate uracil, it becomes thymine, which is present in DNA. It's not present in RNA. So uh, yeah. So I just go back to the structure once. You see uracil and thymine. You just add a methyl group here to uracil, it becomes thymine, right? So yeah, the answer would be DNA, not RNA. Moving on. TRP is tryptophan, by the way. A lot of these. Yeah, the correct answer is one. Just going back here. UGG codes for tryptophan. There's only one codon. Uh, Methionine is also coded by one single codon. You can remember that as well. Do we have any answers? Okay, a lot of D's. Mostly D. Okay. Uh, I'll tell you D is not the answer. I just want you to read the question properly again. I see a lot of A's and D's, B's. Okay. Okay. Yeah, I'll tell you this again. The questions are going to be a little bit worded tricky, right? The wording is going to be a little bit tricky. So you need to read the question properly before answering. So let us go through all the options here, right? So the the answer would be prediction of protein three-dimensional structures. The other options, just try to see here this determination of protein 3D structures. Determination cannot be done by bioinformatics. Any kind of determination cannot be done because for determination you need experimental validation, wet lab validation. Bioinformatics is all about predictions, all about models, all about trying to predict something. Right? You cannot determine anything through theoretical frameworks. Bioinformatics is a theoretical framework, right? You have computer science or statistics, you have other tools at your disposal, but it is a theoretical framework. It is not a wet lab or a experimental framework, right? So any type of determination, you need experimental data for that. So mostly determination would look like for example, X-ray crystallography would be determination of 3D protein structures. And option three, Sanger sequencing. The sequencing itself is not part of bioinformatics. The sequencing is just what you're doing in Sanger sequencing is basically, uh, I'll not go into details of Sanger sequencing, but what you're doing is you're adding base pairs one at a time, and you are trying to see and you're capturing that signal, that binding of one base pair at a time. You're capturing that through some radioactivity or fluorescence and so on, and you're getting one sequence after another. So the whole sequencing procedure is a wet lab procedure. It's an experimental procedure. It's not bioinformatics. The data that you get after sequencing, the things that you do with the data, that would come under bioinformatics or computational biology or whatever, right? But the generation of the data itself does not come under bioinformatics. It is wet lab. It is experimental data. So I hope this clears things up. Do you have any questions? Okay. Okay. So IMBOSS I have not discussed today, but I'll not dwell too much time on this. It's basically the second option, European Molecular Biology Open Software Suite. I'll just quickly give you, I don't know if I have it open somewhere. Uh, This is the home page. They tell you what are the things that you can do about. You can go to about and take a look. What are the things that you can do? Sequence alignment, database searches, protein motif identification, and so on and so forth. So, yeah, this is also a kind of an older software, uh, but this is still updated. You find updates for the software till now. Please take a look if you're interested. Right. Okay. I see a lot of answers. 14, 14, ETA, 15. Okay. So just to be clear, these are trinucleotides, not dinucleotides. Right? Three sets of three. So there's a simple formula. If you want to calculate the number of k-mers possible, k is the number of sequences in the smaller sequence. So basically, in this case, k equal to 3, right? So we call that k-mers. So if you want to find overlapping k-mers in a set of total sequence of, say, n nucleotides, the total number of overlapping k-mers would be n minus k plus 1. If it would have been a dinucleotide, it would have been n minus 1, as we saw in the base stacking energy thing. But if it's a trinucleotide, it's basically plus minus 2. But the idea is n minus k plus 1. These are called k-mers. K-mers is something that you'll come across in NGS as well, generation sequencing. So what they do is they break. So you cannot. The DNA is 3 billion bases long, right? For a human being, you cannot sequence the DNA end to end. You cannot start from base one and go straight like one after another sequence the base and go to 3 billionth base. You cannot do that. That is not possible. So what we do is we break it down into smaller chunks of DNA, and these smaller chunks of DNA are known as k-mers. K representing the number of nucleotides in that smaller chunk. So in most cases, it is like 200, 300 bases long. So that k-mer concept comes there as well. So yeah, just remember n minus k plus 1. It's simple. Oh, some questions regarding k-mers. Um, Faisal, I do not understand your question. K-mers are like what? Like, especially, can you tell me what you mean? I do not understand what you're written here. Uh, yeah, Shalaka, do you want me to repeat what are k-mers or like then minus k plus 1 stuff? Ha. So I can see the analogy. If you consider the DNA as a whole sentence, then the words inside that sentence will be k-mers. That would be uh, the idea. Okay. Uh, yes. So just I'll come to this. Just let me repeat the last question once. So the question is that how many trinucleotides that can be obtained from a sequence of n nucleotides, right? So trinucleotide would be like shifting of like uh, uh, you have a whole sequence. You are shifting by one window and taking three nucleotides together. So if you have uh, so what you're doing trying to find is how many overlapping sequences of length 3 can be found from a total sequence of 16. That is the question you're trying to answer, because what you're doing is finding overlapping sequences, right? These trinucleotides will be overlapping. So the idea is that if you have n nucleotide uh, and k is the length of your smaller set of nucleotides.
Okay, you guys start now.
Sorry, uh, did anyone say it? I didn't hear. Okay. let me go back here. Yeah, so the idea would be to have, so if you have a sequence of length n, and you're asked to find what is the number of overlapping sequences of length k you can find from this sequence of length n, then this would be the formula for that, n minus k plus 1. If you have a trinucleotide, this would be n minus 3 plus 1, which is n minus 2, in this case 14. If it's a dinucleotide, it will be n minus 2 plus 1, which is n minus 1, the one that we saw for the base stacking example as well. So it's simple. It's just if you are asked for overlapping sequences, how many would you get? The answer would be n minus k plus 1. I hope that makes it clear. Sorry, what is this? BODMAS? I do not understand what you're trying to say. First, you have to add then subtract. Uh, it doesn't make any difference, right? If you add and then subtract, like addition and subtraction is interchangeable, right? It doesn't make any difference. Just you put in the values of n and k, you'll get the answer. I'm not sure where you're getting 12 from. If you do it manually, you just try to find, you take a sequence of 16 nucleotides and you try to find the overlapping nucleotides manually, you'll get 14. What can happen is there can be like two sets of these smaller sequences can be same. That is possible depending on the DNA itself. There can be repeats in the DNA and two sets of overlapping k-mers can be the same. That is possible. But distinctly, you'll find n minus k plus 1. Uh, I hope that is clear. Okay, in terms of the relational database, uh, it is basically a, a table, a big table, where you have the data into predefined tables of rows and columns. And uh, since this is a relational database, so one set of data is always connected to another set of data. So you can have different set of these tables, but each table would be connected to another table through some key. Say, for example, uh, um, you have one table as like gene information. So you have a set of 20,000 genes, you have 8 samples, and you have that as a table as gene expression. The second table would be that set of genes and what is the, say, for example, number of variants in that gene, right? So that can be another table. But in these two tables, the common attribute would be the set of genes. So a relational database would have like these distinct big tables of columns and rows, but they would be connected to each other through some keys. So that would be the idea with at least one common attribute. Okay, moving on. I can see a B in the between. Uh, just to be clear, DNA sequences are not. Oh, sorry. DNA sequences are not formed by hydrogen bond. The hydrogen bonding is between the bases in the DNA. So it does not form with this help with the sequence of the DNA. What it helps is keeping the two strands of the DNA together. That is what the hydrogen bond is for. The phosphodiester bond is the backbone of the DNA. The backbone of the DNA is made up of the sugar molecules and the phosphate. So the phosphodiester bond with the phosphate and the sugar molecule, that is what makes up the backbone of the DNA, through which the DNA is formed. Right? So, any okay, a lot of C's. Okay, as I said, what you need to do is since you have like the the hydrogen bonding energy is given like this, the stacking energy is given in pairs. So you need to find the pairs of sequences from here. So first will be CG, then will be GT, sorry, then will be TA, then will be AC, and so on, right? So you have to just add them up, the stacking energies, and normalize it by the length of the DNA molecule, the length of the sequence itself. So uh, just sum this up. -6, -8, -7, and -10. Just sum this up to get -31. And since you're asked for four uh, different pairings, CG, GT, TA, and AC, the idea would be to divide it by the by 4, which is the nucleotide, the number of uh, pairs that are possible. That comes to 7.75 negative. So, and one more thing to keep in mind, the the base stacking energy or the hydrogen bond energy, the delta G will always be negative because it is thermodynamically feasible. Otherwise, the cell would have to spend energy to keep it in that structure or that form. Right. So the, another way to cross check if it is correct or not would be to check if it is negative or not. Right. Because your DNA stacking energy should always be negative because it's a stable molecule and thermodynamically feasible. Uh, any questions? Yeah. Okay. Then moving on. Ah, most of you have got it. Yep. Yeah. So the answer would be schema. Uh, Ontology is basically it's kind of a machine readable way of representing knowledge for a particular domain. So, for example, if it is a uh, taxonomic database, the ontology there would correspond to the different classifications of taxonomy. The schema is basically how the data is stored, and ontology is basically what the data means. And format and all data retrieval, these are like self-explanatory. But ontology, remember, it is what the data means in a more technical, in a more machine readable way. It can be IDs, it can be some words or something like that. And schema is basically the structure of the database itself, how the data is stored. Okay, ontology is the what, and schema is the how. Questions here? Okay, this is also very simple. And the answer would be two. Yeah, sorry. Yeah. Okay, I think we've discussed all the 10 questions that were there. I think for most of the weeks for this particular course, it is 10 questions. So I think, yeah, I'm done from my side. If you have any questions, you can ask.
Sir, can you explain RevSec sequence and forward and backward reading frame?
Okay, forward and reverse reading frame. And what is the other thing that you said?
The RevSec, professor said something about RevSec.
Resic.
Yes, REVSEQ.
Oh, Resic. Okay. I don't think I have put it here. Okay, I think Resic is the database, I think, if I'm not wrong, for reversing uh, the nucleotides. It's an embossed database. Yeah. Yeah. I think you can put a sequence here. So if you give the sequence, what you can find is basically the reverse sequence for that. So the sequence that you are putting is in a 5' to 3' manner. What will be the 3' to 5'? The complementary, the reverse sequence for that, right? The complementary sequence would be the sequence of the complementary strand itself. And yeah, I think RevSec does this. And the forward and reverse the reading frames is basically similar concept. So this is the open reading frame that we discussed, right? Yeah. Where can I put this? Yeah. So the open reading frame, this is the forward open reading frame. Why is it forward? Because it is starting. So it is not given here, but it is given here. It is 5' to 3'. You're reading of the open reading frame is always 5' to 3'. All the enzymes in the cell, be it for transcription, be it for replication, be it for anything, it reads the sequence from 5' to 3'. So any sequence in that direction is the forward frame. So this is the open reading frame. Generally, when we talk about the open reading frame, we talk about the forward frame. So it starts with the start codon, right? The forward frame would start with the start codon. The reverse frame would be the same sequence for the complementary strand, right? So if you say, for example, when this comes into handy when you are sequencing, right? So when you're sequencing the DNA, uh, so you every DNA will have like two strands, right, with the same information, just one in a opposite direction, anti-parallel strand. So you do not need to sequence both the strands together, right? If you sequence one strand, you know the sequence of the other strand. So in some cases, what can happen is instead of sequencing the forward strand, the 5' to 3' strand, you have sequenced the 3' to 5' strand. So you have the same data, but in a kind of an opposite manner. So you have the reverse reading frame. you have to convert it to an open reading frame to get a better understanding of the data itself. But these are like kind of terminologies which are telling you in which direction to read the sequencing. If it's a forward reading frame, you read it from 5 prime to 3 prime. If it's the reverse one, you read it from 3 prime to 5 prime. Right. So that is the idea. So these are all terminologies which are telling you, these are small nuances in terms of the sequences, like in which direction the sequence is reading and in which direction you should read the sequences. I think that that is all in terms of forward and reverse. For this particular thing, I also the RevSeq thing. I have not used this thing. I'll be very honest with you. But if you if you find any issues with this, you can drop me an email. I'll take a look of over this. But I think this is quite simple. You paste a sequence in a FASTA format or some format, and it can reverse complement and so on the sequence itself. So I'm not. I don't think this would be a problem, right? I think there are command line arguments also given. A lot of options.
Back to me if you face any issues in this thing.
Okay, sir. Thank you, sir.
Okay, upstream and downstream is with respect to where you are in the DNA itself. So, for example, you are talking about upstream and downstream of what? Upstream and downstream, we generally say in terms of a gene or a region of the genome, right? So, if there's a genic region of the genome, you are saying that there is some mutation upstream of this region, right? So, what upstream means is towards the 5 prime. Towards the 5 prime end, from a particular region is called the upstream, and towards the 3 prime end from a particular region is called the downstream. So downstream and upstream are with respect to a particular region in the genome. That can be a gene, that can be some particular region of interest or anything like that. And upstream and downstream are defined on the basis of that. Right? So upstream would be towards the 5 prime and downstream would be towards the 3 prime. Does that answer your question? Okay. Any more questions?
In the initial slides, you talked about two distances, like the very basic parts, like between two bases or maybe something. I just could not follow that part.
Okay, just a minute.
Like something 0.34
Oh,
nanometer.
okay. Just 1 second, let me share my screen. This thing you're talking about? This thing, right?
Yeah, this one.
Yeah.
Yes.
Yeah. So this is basically a simple thing. So you have two complementary strands of DNA. Now this DNA is not in a ladder form, right? So you have a set of bases on this side, a set of bases on this side, and they are connected by hydrogen bonds, double or triple, depending on AT or GC, right? Now this is a ladder kind of a structure. They twist this ladder structure in a right-handed manner, with a certain distortion to form this helical structure of double-stranded DNA, right? Now, in this double-stranded DNA, to form this helical structure, what happens is you have different grooves in the DNA. So you have these major grooves, which are like bigger indentations compared to these minor grooves. These are the minor grooves, right? These are like smaller indentations, but these are complementary. If there is one minor groove here, there will be one major groove on this side, and if there is a major groove here, there will be a minor groove on this side, right? So the distance is based on, basically, if you take a distance between one major groove and the next major groove, so this would be the distance. The distance approximately comes to 3.4 nanometer. And in this stretch of 3.4 nanometer, there are around 10 bases on an average that are paired from the two strands, right? So you have 3.4 nanometer and you have 10 bases on average in the 3.4 nanometer region. So on an average, the distance between two bases would be around 0.34 nanometer. So this is how generally the structure of the DNA would look like. And you can also calculate distance between two minor grooves as well. So from here to here, it would be the same 3.4 nanometer, or two different other major grooves from here to here, say for example. That would also be 3.4 nanometer. I hope this answers your question. Huh? Thoughts?
So from the right hand side, it is the major groove, or the left hand side, it is the minor groove, something like this.
Yes. So you see the indentation here, right? This is the major groove. On the same plane, if you look at the left hand side, this is a smaller indentation. This is the minor groove. So if you have a major groove on the right, you'll have a minor groove on the left. If you have a major groove on the left, you'll have a minor groove on the right. It is just how, if you just take a ladder, kind of spin it, like distort it, make it a helical structure, physically this is how the helical structure will look like.
So as we say, A DNA, B DNA, some are
Hmm.
right-handed and some are
Some are left
left-handed.
hand.
So it
These
is
are
just
mostly
based like this.
this is the right-handed helix. I think right-handed is the BDNA, if I'm not wrong. BDNA is the one that is the most common across organisms, and that is the one that we mostly study. There are other forms of DNA, A and Z. Some are left-handed, twisted. Some are some have some other modifications, but we mostly focus on BDNA, if I'm not wrong. Please correct me if I'm wrong. I think BDNA is the one that we are talking about, where it's a right-handed helix, and with this kind of a groove structure.
So how do we understand, sir, that this is right-handed? That means major part will be on the right-hand side.
No, the so it is very difficult for me to make you understand in terms of, like, you have to imagine it. If you imagine this as a double-stranded DNA, and there are bonds in between, so this is kind of a ladder-like structure. So you can twist it like this, or you can twist it like this. So the twist is towards the right hand side, and how they find it is basically through these X-ray crystallographic methods. They have so I am also not very sure about the experimental procedure, but they uh, kind of find where the grooves are and in which direction the major grooves are pointing at. So from that information, you can find out if it's a right-handed or a left-handed bird. Structurally, I'm not very sure about the experimental
Yes.
methods. There are a lot of experimental methods by which you can find, but this is not very difficult to find. Like, you can, at least standing today without all our technology, you can say if it's a right-handed or a left-handed twist.
Thank you, sir.
Okay. Okay, I have a lot of questions here. Let me admit. Okay, what is the primary difference between global and local alignment? Alignment will come to, there is a whole module on alignment, will come. Will There are a lot of kind of numericals as well of how we calculate these alignments and so on. I'm skipping the alignment question because we'll discuss in details all the kinds of alignment, local and global as well. KEGG and OMIM are different types of databases. These are kind of these do not store any basic information, any primary or even secondary information. It kind of, you can think it of as tertiary information. So OMIM is a database where it stores basically uh, the 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 diseases that are associated with particular genes. Say for example, TP53 is a very well-known transcription factors which is involved in cancer, different types of cancer. So if you search for TP53, you go to OMIM and you search for a particular gene, and you go there and it will tell you all the phenotypes, the diseases that are associated with this particular gene, and what are the modes of inheritance, dominant or recessive or so on and so forth. In some cases it can be somatic as well, not germline. So yeah, this kind of information is present in OMIM. You can also have information in terms of the different variants. So it also kind of tells you about different functions of this particular protein from different literature. So they cite these literatures. There's a lot of TP53 is a very well-known protein. There is a lot of information on TP53. I'm just trying to find. You also find different information of variants. Just let me. TP53 is probably not a very good gene to I have to scroll a lot. Just a second. Huh. So they quote studies for which this particular gene has been studied, different variants. So E4 allele is a particular variant of this gene, ApoE gene. This is a very important gene in Alzheimer's, in lipid metabolism, and so on and so forth. You go to allelic variants. These tell you. So this is one variant. At the 158th position, the arginine has been converted to cysteine. This is the these are the RSIDs, the mutation IDs associated with these particular variants, and it tells you when this mutation happens, what happens to the individuals or mice or humans, whatever is there. So this kind of information is present in OMIM, is what I'm trying to say. If you have a gene, you can find out all the different types of diseases that gene is involved in, different types of variants of that gene that has been studied, and so on and so forth. So this is OMIM.
Sir, OMIM is a primary or a secondary uh,
It
data
is not.
like
Uh,
database?
it is very hard to classify OMIM. I think you can put forward an argument for it to be. I am not very sure. Just let me search. You can put forward an argument for this to be a primary database, or also you can put forward an argument for this to be like a kind of a tertiary database. Um,
It curates and summarizes information derived from primary biomedical literature and
yeah,
other raw data
yeah, yeah,
sources. So,
yeah, yeah, yeah. So it is not exactly secondary as well. So it is kind of tertiary if you look at it in that way. But you can also look at it in for the fact that it just curates information from literature, that this gene associated with this disease, this gene associated with this disease. So that is also secondary database as well, right? So I don't think OMIM. So OMIM and all these cake kind of cake is a molecular pathway database. It's okay, but I am not sure you can classify very well. I'm not sure. It is probably possible. It's a secondary database, but you can also make a case for it to be a tertiary kind of a database, because all the information that it is having is also coming. A lot of it is also coming from secondary database as well. So these variant informations and these kind of things, these are coming from DBSNP or databases like that. ClinVar is another database for clinical information. So it is kind of secondary or tertiary database as well. You can make a case for that. I'm not sure about OMIM. AI says it's secondary, so probably it's secondary.
okay, sir.
Yeah. And someone asked about KEGG. KEGG is a similar kind of a database. KEGG contains all the pathway information, the genes and the proteins involved in different pathways. So you can classify it as a secondary database. Standard steps for analyzing NGS data. This is a course in itself. I can't talk about this in like 5 minutes. But I think we'll be covering a little bit of NGS there, probably, if I have the.
Thomas, sir, what you are working on? Like we do not have.
Okay, so I am I am a PhD student, third year. I am in ISC, so I work at the Center for Brain Research in ISC. So here we work on genomic data from Indian individuals. We study mutations. We statistically associate these mutations to different phenotypes and diseases, and then after that there is a lot of downstream steps as well. I'll not go into details of that, but my major work would be to use sequence data to understand mutations and their biological implications, and also at a population level how these mutations are distributed and so on and so forth, for different types of diseases. This particular center that I'm working on is focused on Alzheimer's, Parkinson's, neurodegenerative diseases like that. So
Okay,
yeah.
sir. Are you from Kolkata?
Yep.
Sir, me too.
Oh, good to know.
I am Bangali.
Oh, ভালো ভালো. Okay. I'm sorry, Sonam, I can't help you with internships. Uh, the thing that I can say is like you can keep on mailing people. That is how you get internships. Bioinformatics related jobs. Yeah, some level of coding is required, but it's not a it's not something that you can't learn in a certain small period of time. So it's not like very high level coding or anything like that. It is basic coding, but you need to learn any one language, R, Python, or so and so on, or shell scripting or so on. Do not learn Perl and what was the other language? Basically, the older obsolete language. Don't learn those. Either learn R or Python or something like that. Computational neuroscience is not. I don't think this course will suffice to that. It's a completely different thing altogether. My education, I have done my BTech MTech dual degree in biotechnology from NIT Durgapur, and after that I've joined here. What is AMR? I'm sorry, I'm not sure for pathogen. Oh, see, I do not work with microorganisms, so probably I'm not the best person to ask this. But probably those metabolic pathways, and I think there will be microbial resistance, like this basically antibiotic resistance and this kind of things. There will be databases for that. I am not very sure because I do not work in this particular field. Start with anyone. Python or R is very similar. But if you're going more into statistics, I'll say R. If you want to learn general coding more, I'll say Python. I think that is all. I don't think I've missed anything. Okay, then I think we are almost there for 2 hours. I think I'll close this session now. No more questions.
Thank you, sir.
Okay. Thank you guys.
Thank you.
Thank you for joining. See
Thank
you
you, sir.
next Tuesday, 6 p.m.
Thank you, sir.
Thank you.
Thanks, sir.